The next session should be presentation 3, annotation. Yeah, then I would like just to hand over to you. It's your session and yeah, your moderation and discussion together with the panel members. So
thank you very much.
Please start. Yeah.
Thank you very much. Yeah, so welcome everyone back to presentation 3. We are going to speak about annotation. In particular, we are going to speak about problems and solutions that we have encountered in different European projects for actually be able to standardize the process of data annotation that we know that contains lots of issues, and there's a lot of variability in this process itself. So for this, first of all, okay, so I know that the speaker is not here yet. I can talk briefly about a few topics we have been discussing in the meantime in the AI for Health imaging working group relating to data annotation, and hopefully we give time to the speaker to connect. So some of the topics I think I would like to cover also later with the panel members would be regarding, for instance, the current challenges that we have been facing. So we have identified a few of them. So some of them, for example, regarding the data formats that were how we are actually saving and recording or annotations. And for this, we have actually divided the topic in 3 main aspects. So because it's different if the annotation is a detection problem or a classification or a segmentation problem. So we also can actually go further and discuss what we think are the differences between what the detection task is, the classification task is, and what the segmentation task is. But for each of these ones, the format, how we are storing the data is different. So another of the topics I think is very important is about the image, both image and metadata quality and harmonization. So it's very important, I think, to try and set some sort of boundaries and guidelines for actually be able to identify good quality image and metadata. And when it's not the case that this data complies with good quality standards, identify methods to actually harmonize this data. So we will also probably discuss this further. Another of the topics is image quantity. So we know that we have weak and scarce annotated medical images, and yeah, sometimes also lack of balance when we have different classes. So when we annotate, we might have lots of images that are related to a specific class, but very few that are related to another class. So these at the end can lead to many issues when actually we are performing data modeling afterwards. So probably strategies for data augmentation in this case are very important. Also, there's another big box of issues that regard for in my understanding to the human-based biases that can arise when we are annotating imaging data. So we can then discuss about how we measure inter-observer variability of these human processes and observation, if we have to take into account previous knowledge, experience, personal skills. Also, how sometimes we annotate from radiological structure reports or from radiological reports that have no really a very good structure, and it's all free text on them, and how we actually select and annotate our data based on these reports. And then also we can discuss what we always say was the difference between gold and silver standards. So basically, yeah, what we consider to be or gold standard, something that we really know certainly that is true, or a silver standard, something we consider sometimes that is true, but of course it can have some biases because of the process that we follow to obtain this ground truth. And also there's another box regarding metrics. So for metrics in annotation, again, we should probably discuss about the different tasks. So it's not, we don't use the same metrics depending on the type of annotation task that we are performing. Is not the same. If we're performing a segmentation task, we probably have been using DICE or Jacquard metrics, or Hausdorff distances. But if we are actually performing detection or classification, we might want to consider probably other metrics, maybe more regarding F1 scores, or maybe areas under the curve of the ROC curve, or others. So we can discuss about what we think could be the most suitable metrics for assessing annotation quality. And finally, it's also a very important topic, is basically AI-based methods that actually present challenges, or we face with different challenges when we are applying AI-based algorithms to try and perform annotations. And so some of them are really relevant in the field of medical imaging, and not only affect annotations, but other sorts of models. But in here in particular, we have seen problems regarding model generalizability, overfitting, problems with the universality of our models, how we monitor the performance of our annotation algorithms. So this is very important in terms of, like, we have a big power here, where actually we can deliver probably models for annotating that can be validated pretty much in real time, as for instance, there's a new image performed in a hospital. So that would be a very nice way to actually annotate them directly in the systems, and then that they can verify, they can actually edit or validate these annotations. So it's a very important topic, how we actually do this monitoring for the performance of the models. And in here we are introducing the specialist radiologist in this process. So this would be some of the topics I had thought could be very interesting to discuss. Okay, so I can see here, sorry, that the speaker might not be available. Okay, so I can just, since I had been discussing a little bit already, if the other panel members are available, we can discuss some of these topics, or some others. Yes. Would that be okay? We can try and discuss some of the topics, and maybe talk about the different strategies that we have been following in some of the European projects that we are working on.
Sure, let's do it.
Okay, thanks. So we have here Clemens.
Hi.
Hello. And then also we have Martin.
Yes, hey.
Hello. Okay, perfect. Thanks a lot for joining. I've been speaking a little bit about different, aspects that we have been facing and problems we have been facing. So feel free to just comment if you have any input on any of these, or if you just want to speak about your experience, for example, in EuCanImage or any other of the European projects. Thanks.
If you want, I could start. So I recognize a lot of the problems you discussed. We had almost everything as well in EuCanImage. I think maybe one important part is the balance between sort of the optimal annotations and the time that people want to spend on it, because I think we have approximately 25,000 cases. and the radiologists are not going to fully annotate them. So it's a trade-off how you do that. So what we now did in the project is now for a part of the patients, we will sort of do full annotation. So at least for a part of the patients, we have, let's say, the best annotations we can have. And then for another part of the patients, we've decided to only do a sub-part of the annotation. So for example, for some patients, we have full annotations, so pixel-wise annotations of all the tumors we could find. But for other patients, only a couple of tumors, or we go for bounding boxes, for example, so less time-consuming annotation. So per use case, we balance a bit between what's optimal and what's pragmatic to say.
So at the end, thanks for this. So at the end, basically for the different use cases that you might have in your project, you are not annotating all of the data. You're just annotating maybe a few % of this data. Is this correct?
Yeah, so we sort of have two strategies indeed. So for part of the data, the perfect strategy, and for part of the data, let's say the minimal strategy, so that we can still on the other data sort of validate whether some parts work. But it's impossible to annotate everything.
Mm-hmm.
Yeah, it's indeed very challenging. Yeah, so we are doing we're doing various projects where we indulge in hybrid imaging annotation. So one project is with Professor Gatidis from the University of Tübingen, where we annotated FDG PET-CTs. Yeah? And actually, Sergio's group uploaded an annotated data set to TCIA, and we started a challenge. It's called the Auto PET Challenge. So auto like car, yeah, Auto PET Challenge. If you want to check it out, it's online. It's really interesting, and we did it together with MICAI, yeah, and both societies, so the European Society of Hybrid Imaging and MICAI endorsed this challenge. We're going to present the results on a special session on it on the European Congress of Radiology 2023. Yeah. And we are evaluating basically automatic segmentation algorithms versus human performance. So that's a story we've almost tackled. Patients with lymphoma, melanoma, and lung carcinoma were integrated. So that sounds kind of feasible, yeah. But of course, the very likely idea is to also integrate, for example, PSMA PET-CT, yeah, and do something similar with PSMA PET-CT. And you probably know that patients that are, for example, evaluated for lutetium PSMA therapy are usually patients that do not have one or two metastases, but are usually metastasized throughout the body. So we are evaluating also this group of patients, and we are undergoing heavy annotations in PSMA, PET, and CT, and this is really gruesome. Yeah, I do have to say we are using a certain software that helps us to perform this task and to generate a very homogeneous and very standardized data set that we will also make publicly available, yeah, in a very similar structure. Yeah, but I completely agree that the time you have to invest, the time and love you have to invest to do this properly is painful, and I also do the cross reading. Yeah, so I indeed I'm physically involved in it. Yeah, so that's interesting, but I guess it's a very important job to do. Yeah.
Yeah, regarding what you were saying, I had the question of asking actually, how did you compare? So this you said you were comparing human versus automatic annotation. So how this comparison works out at the end? What are your results? What have you seen? Because so far we have seen many times that actually when we are actually developing a model with a lot of data, at the end the levels of variability that we observe between human annotators and then human versus a machine are very similar. So that means that actually there's also a big variability in the humans when you are actually studying different radiologists, in this case, who have different experiences and skills to perform the same annotation, in particular when the tumors are or lesions are tricky, as you're saying. So how have you seen these comparisons in your studies?
So I completely agree. The inter-observer variability in the annotation plays a very important role. And also when you do it twice, or if you, for example, evaluate PET-CTs before and after therapy, it can be, it's very hard to standardize this from a human perspective. The results of the AutoPET challenge are not completely available, but the interesting thing about it, of course, is that globally a high number of groups handed in or evaluated the data by their algorithms. And this will be particularly interesting to see how the variability of the performance is DICE as a target parameter, of course, and if there are true differences in the performance. But I think what you were alluding to was more the inter-reader variability in the ground truth. So the human performance. So the way we do it here in Munich is that we have at least 2 readers, and usually one is very experienced. So at least 5 years plus of hybrid imaging experience, double qualified in radiology and nuclear medicine. So it is very difficult. But we are as the results become available, this will be very interesting. And of course, we are also handing out a prize for the first 3 top performers, and this is going to be very interesting. But in general, I think what you were asking also was the question, subhuman performance, human performance, and suprahuman performance. And I think what we see very generally is that we are just not ready technically to see frequently a superhuman performance. I think we are all longing for this, because I think we are all enthusiasts, and we know it's going to be possible, but I think at best, we are currently seeing human performance standards, and this is not only true for academic or scientific research tools, like auto segmentation, but it is also true for clinical AI algorithms. We are using a whole set of AI algorithms for reading support here at the University of Munich. And of course, basically the algorithms support you, and what they give you is basically human performance. They are also in their clinical application, usually not in a superhuman performance spectrum. I don't know how your experience is, but that's what I see research-wise, at least for the most part, and clinically in any case.
So maybe one story from one of our studies. So in EuCanImage, we also work on colorectal liver metastasis on CT. And previously we did a study where we asked 5 radiologists to annotate the liver metastasis. And we saw that the overlap between the radiologists was only approximately 70%. They simply didn't agree on which lesion and which pixels belong to the lesions. And also they missed actually 20% of the lesions. We had to go back to the pathology reports and point them out that in specific segments of the liver a lesion was found. And then they could find something that looked like a lesion. But this raises the question, like, what are lesions, which pixels belong to the lesions, and what's a decent performance to get? So also for an AI algorithm, getting an overlap of 100% with one reader, well, is that really accurate, or is that only mimicking a specific reader?
Yeah, I completely agree, and I think this is a very interesting aspect. But there's another aspect I just thought of, so I would like to go one step back. I was when we were discussing the hybrid imaging approach, and the accuracy of the segmentation. A lot of times, segmentation is performed only on PET, or only on CT. And what we are seeing right now, when we segment the contrast enhanced full diagnostic CT side by side with the PET, is how large the variability is between the size of the PET segmentation, and the CT segmentation. it's incredible. Yeah, it's certainly true for PSMA, and it's certainly true for the somatostatin receptor ligands. Yeah, it's very interesting. So I think it opens up even a completely other field of discussion about the accuracy of the human segmentations as the reference standard of our AI segmentation algorithms. Yeah.
I completely agree with both of you. So that's a bit of that when we have been always discussing, this is not really a gold reference, it's more like a silver reference that actually, yeah, you're using it, but it can be wrong as well. And probably it's very important at the beginning to establish what is normal and what is abnormal, considering before you are going to annotate. I know that sometimes radiologists think, I know very well where is the lesion, but yeah, like we have seen the same thing, that afterwards they have big discrepancies, and then they have to discuss about it to actually get to an agreement. So yeah, I think yeah, it's a very important topic. Another topic that I wanted to discuss with you is what is the best format to store annotations. So basically now there's been a big, for sure, when we are developing models, seems to be like, people enjoy like storing data in like nifty formats or formats that are more easy for treatment afterwards, for processing. But the current as well is to actually try and store this segmentation, for example, in particular, if it's an image segmentation, in the DICOM format, which is probably the format that is more standardized for medical imaging. So basically maybe storing it in the DICOM seg objects. So we have been using these objects to actually store the segmentations that are associated to a specific DICOM image. And but I know that in the field it still has, there's some controversy about what should be the standard format, or what everyone should use to store these annotations. I don't know what is your experience in your projects or in your own labs about how you store annotations.
Yeah, should we have one format? That's maybe the question, because I think in our project, at least the segmentation program that we use, so the collective mind suite uses RT-struct, but then as soon as the segmentations arrive, we do several conversions. Because we know that some people in the project like with Nifty, some like with another format. So in the end, we provide them in multiple formats, basically, so that any user can pick what they want.
Okay.
I think, well, I fully agree. In a way, we need to find a joint format that everyone uses. I think this is the main message. But I think what also needs to be discussed is if what you said, Leonor, if the segmentation, what's the value of having the segmentation stored with the DICOM image? Yeah, is this a higher value actually when you export it? But it takes forever, at least with our system. Yeah, it takes forever if you export all the images with the segmentations. It's really a trouble, but we think that there's a higher value in it, certainly if you're preparing the data sets for public access.
Indeed. So yeah, like another one of the topics that we have been encountering, for example, in our projects, in particular in Preimage, has been in the process of when we have the data, the data is downloaded, we have it in our servers. Before we annotate this data, we are encountering lots of over anonymization of this data before we actually can go and annotate it. So in particular, we are not talking only about annotation of images, but also annotation. For example, we were discussing about this model for annotating in MR, for instance, what the different sequences that we have. So when we have an MR study, there's plenty of sequences, T2 weighted, T1 weighted, diffusion, perfusion sequences, flare, etc. So basically, to try and put annotations on these sequences. So this is one of the models we have developed in the project to try and facilitate and automate then the analysis of these sequences for extracting biomarkers or for developing models. So one of the issues we encounter is that in the anonymization processes, there was a very aggressive way of anonymizing sometimes when the centers were sending their data, because this anonymization comes from their own hospitals and sometimes we cannot really control it. And they were deleting more information from the metadata that probably then we would need for maybe annotate this sequence or this data set. So yeah, at the moment we have been facing this. We have been working on building a model for annotating automatically sequences that do not have many of their metadata fields. Of course, we still need to have, I don't know, like echo times or flip angles or repetition times or the orientations. So I don't know if you had been facing a problem in trying to also when you have lots of studies, image studies, to try and annotate them, or yeah, like, and what challenges have you been facing with this? So at the end, in your project, probably you want to have a pipeline that does all the image processing automatically, and you don't need any human intervention, ideally. So how do you face these sort of challenges that can arise when you are trying to automate this process?
Yeah, maybe I can chip in 2 cents from my side. Of course, we are dealing in our studies usually with different single-site partners. Yeah, so we are not accumulating data. So
Right.
yeah, the perspective is somewhat different. But I can actually contribute from a current project. Yeah, where we are trying to get an external test data set. Yeah, and we are actually facing big troubles with the anonymization, and it is on the one hand heterogeneous. Yeah, so obviously the external center, 2 people did it. Yeah, so a part of it is anonymized in one way and the other one in the other way. Yeah, so it doesn't fit together. And it pointed, of course, out to us how difficult it can be. On the other hand, we had another project where it worked very easily, but we discussed it very openly in the beginning. Yeah, so it taught to us that when we are in a situation like this, we hand out basically an information sheet, where the most important points are basically set out. But I know that's not the really core of the discussion you were raising.
Yeah, I think for us, we've not collected all data yet, but I'm sure this issue will pop up. And that also was raised in the previous panel session. For sure, there will be some private text deleted, which we will need. But we've really done our best to discuss with all the clinical partners to see if we can keep as much as possible. And during the annotation process, they will at least annotate some scans. So to point out, this is this contrast phase or this is that. I guess it will still be perfect. So what we also tried in the past is to purely based on imaging information, classify the sequences basically. So there we actually use an AI model to classify brain MRI images purely based on the imaging information, because the dye contexts in the end were not reliable enough because everyone was using different anonymization, etc. And that worked relatively well. So yeah, I hope we can do something similar in here. Well, I hope we don't need it, but otherwise maybe we might do something similar.
Okay. So I wanted to also talk to you about, so in the AI for Health imaging working group, we have been discussing with several of the members about using this active learning approach that now everyone seems to be talking a lot, in particular because, so this consists in basically saying that until now, all the models that we build are like, we have a data set, we build a model, and this model can be deployed and can be used. But of course, this model, probably with time, we see something that we call data drift. So basically, it's like the data changes with time and you need to update the model. It doesn't last forever. So basically to try to fight against this, there's this new method or strategy that's called active learning, that basically is like, okay, instead of having a static model that has been only trained with specific amount of data, we can actually be training continuously a model with the data that keeps coming continuously in the hospitals. And segmentation, we thought in the working group, there was a particularly nice task for this, because it's something that, you know, we have continuously new images coming into the hospitals every day, and it could be a tool that could be integrated in the workstation for the radiologist, and then they could automatically actually validate a new case that just pops up in their screen, and they could just say, okay, this segmentation is good or not, and give feedback, and then the model could continuously be evolving with time. So yeah, this is a very, probably at the moment, a very optimistic approach, but it's something that probably these projects like EuCanImage or all these other European projects can actually offer at the end of the project, or probably in the long run for the radiologists, for instance. So do you think this could be something that it could happen in practical, in clinical practice eventually?
Would you like to answer? There's somebody raised their hand. No.
Yeah, probably Joanna, if you want, you can discuss.
Yeah, hi. So I think that in order to approach this very interesting idea, we don't need only to have to have only the correction of the segmentations. We need to know what are the characteristics of these images or this data set that where the algorithm goes wrong. If the algorithm goes wrong in images of this vendor, in images of some subcategory, or it is a random error. So I guess we need to be careful about dealing with this data drift, because we need to know the characteristics of the data that are drifting, I guess.
So I guess your proposal is to study better the data that is coming and see what is the bias or the differences with the data that we have used to train the models.
Yes, to take this also into account, because otherwise, I mean, it is also possible to lead to an updated learning that is wrong.
Yes. Yes, indeed. Yeah, it could happen. Yes.
I think in EuCanImage we're going to do this anyway, because like I said, we will only do the perfect annotations for a small data set. So then in the rest of the data set, we also intend to use the model to sort of generate full annotations. But indeed, if this won't happen, it might be that we add new images. But I always find this very dangerous, because indeed, if you add new images to your model or new segmentations to train, you effectively change the model. Yes. So you have to make sure that it still performs in a decent way, etc. So in research, I guess it's still okay, but especially if you go towards deployment, that's of course pretty dangerous if you just change your model overnight. So yeah.
Maybe there's also a clinical aspect I can contribute in this, because if I understood it correctly, the proposal is to use basically clinical routine segmentations or clinical routine reading to supplement existent and trained algorithms in order to reduce or prevent data drift. Yeah.
Yes.
So effectively in clinical routine, what happens is, particularly in metastasized patients with a lot of manifestations, is that we judge not all lesions. We virtually never segment all manifestations, yeah. But we read the case, and in the end of the day, we need to transfer the clinically relevant message to the oncologist. Is it the same? Is it worse? Or is it better? Yeah. Broken down to the hardcore, yeah. That's what the oncologist wants to know, because he wants to know, can I leave the therapy as it is? Do I need to change it? Yeah. Or can I maybe size it down a little because the patient has a very low tumor load or has responded perfectly. Yeah. Using real world reading data would require a very high level of training and sensitization of the readers in order to provide the quality, what Martin touched on, the quality of segmentations you need. Otherwise, you will potentially spoil your very carefully read training data set, that is annotated by pros, with a high level of dedication, and supplement the training of your algorithm with segmentation data that's either not complete, or maybe not performed in the way it should be, because the clinical information you're extracting is very different from the information you need for the clinical data science approach.
I agree with what you're saying. I think, it's a very, like interesting approach, but probably we will need to be careful on who is annotating this new data and how, and if it's not gonna spoil the model actually. So I think, as you're saying, probably a lot of training would be required also to do this properly, to actually have good ground truth for keep training the models, etc. So it has its own challenges, definitely.
Can I make a maybe controversial proposition on this aspect? We've multiple times said that it's nice if you have experts to annotate your lesions, but I would actually argue that often the experts are the worst, not because they're less knowledgeable, but because they're senior and they're short on time. So if I ask a senior radiologist to do some annotations, they don't have a lot of time. So they usually do it quickly and therefore a bit sloppy. While if I ask a fellow, they usually are a bit more dedicated or they want to prove themselves and they are much more accurate. And of course, this doesn't generalize well, it's not a broad conclusion, but it's not always that the expert is better than the junior, I would say.
Joanna?
Yes, but then I guess there is also this question on how much accuracy is needed for a clinically relevant answer. I mean, you know what I mean. If you miss in your algorithm a few pixels, you will get a lower accuracy. But we are not at all sure if this really is important for the decision. So.
Yeah, no, definitely. That's another important aspect. You need to define what kind of annotation is sort of sufficient for your method.
Yeah, also like if it's an algorithm that already delivers to you the different, I don't know, the different lesions, probably maybe the radiologist would only, as you were saying before, you would only look at maybe the bigger one or wouldn't annotate all of them, would just focus on probably what they usually use for actually passing the information on for the oncologists, etc. So, but if there's a tool that already is delivering and is providing you with all these lesions, and is giving you more information than maybe what you would only look for, that could also be helpful for them. So the other way around, it's not like they need to give us all the lesions, but if there's a model that can provide to them all these different lesions, and maybe they wouldn't consider if there's many, maybe that information could also be useful for clinicians, for radiologists, or not really. they are happy with what they are doing in their own work. I'm not sure. I'm not a clinician myself to answer this question, but like if there's a tool that could give you more information than what you usually would look, if there's many lesions or stuff like that, I think could be useful for them as well.
Yeah, so I guess it depends on what clinical question you want to answer. Yeah, and I pointed out to you in oncology, it's really that easy. Yeah, it's really, is it stable, is it more, or is it less? Yeah, because the oncologist wants to know what therapeutic consequence is coming from this information. And for us as radiologists, yeah, we have one prime requirement. It cannot take more time than it does when you do it yourself. Yeah, and we have seen this with the implementation of AI algorithms in our clinical routine. Yeah, be it lung nodule detection, or be it automatic fracture detection, or be it automatic orthopedic measurements of the hip or of the spine. Yeah. Every time you tell a radiologist to send the pictures, the images away himself to the server, yeah, no auto routing, your implementation process will be a disaster. Nobody will use it. Yeah, every time you tell a radiologist to open a separate program to start the algorithms, the implementation will be a disaster. It's not gonna happen. So even more important than additional information you may be able to add is an adequate workflow integration. Yeah. So if it's auto routing, if it's plug and play, I go into my list, in my work list, I click on the patient and the results are there. Fine. Yeah. Great. Thank you very much. Yeah. I'm willing to use it. Yeah. But if I have to use 5 minutes more, yeah, to fulfill the task, the radiologist for the greater proportion will not adapt it for clinical routine. And that's a very, very bold and very open message, but
Yes.
that's how it is. Yeah. So I think the eagerness is great. Yeah, and people are very interested, but it needs to be a plug and play solution, and the results needs to be clinically relevant. Yeah, those are the two aspects I would see from a very basic clinical radiology perspective.
Yeah, I agree with you. And probably then we could discuss what is clinically relevant, as we were trying to discuss before, like what is the threshold for something that for you could be clinically relevant?
Anything that changes therapy. Yeah,
Right.
anything that has an influence on the therapeutic decision is relevant. Yeah. And anything that cuts your workload is relevant. Yeah. So if you have or increases your quality significantly, yeah, increases your diagnostic confidence. So let's say you have an excellent algorithm for fracture detection, yeah, for automatic fracture detection on a plain radiograph. Yeah. I think this is a solution that will or is received with open arms, because every radiologist wonders when he or she looks or at the image, didn't I maybe miss it still? Yeah, or maybe I can ask a colleague. Yeah, and now you have this digital colleague. Yeah, so this is very comforting. Yeah, it increases your diagnostic confidence, so it increases the probability that it's used. Yeah. Second thing was the workflow, and the third one I forgot.
But I think you touch indeed upon a very important point, that performance alone of such an algorithm is not enough. Indeed, the usability is highly important. So in EuCanImage, we're developing further these future AI guidelines on development, indeed on deployable algorithms. And usability is one indeed of the guiding principles, because indeed it has to be properly integrated in the clinical workflow. You have to provide proper training to people, but people also need to feel confident, satisfactory. And you need to monitor as well if people use it and if it actually helps them in their decision making as well.
Yeah. So let me build one bridge of the auto segmentation. Yeah. So the whole body auto segmentation tool. Yeah. So what's the idea? What could happen? Yeah. For example, one clinical use case that would be quite interesting is an automatic assessment of total body tumor volume. Yeah. So, because what we are seeing with the more and more effective anti-cancer therapy is that in some tumor entities, cancer is becoming a chronic disease. Yeah. So there are two things that are interesting and can be very cumbersome in patients that have just this totally spread metastatic disease. Yeah. Is that you need to check, is there a variability of total tumor volume? Yeah. Of course, when you think about the PET images, black and white, this could work quite well to assess the vital tumor volume. Yeah, but in general, having a parameter that says, well, automatically you open the case and it says the total tumor volume of this patient has remained the same, yeah, since the last exam. That could be very helpful because it gives you the primary assessment of the disease. And then there's one more aspect, and that also comes into this clinical situation. What we are also seeing is an increased tumor heterogeneity, an increased metastatic heterogeneity. That means that some metastases behave biologically different than others. Yeah. So imagine you have just one metastasis that is coming from a different cell clone and has other mutations and starts running away, starts to grow. Yeah. And you have to look at 150 metastases. That drives you crazy. Yeah. So total assessment of total tumor volume and an automatic pinpointing of lesions that have significantly grown or decreased in size, that would be an application I would regard as clinically helpful.
Joanna?
When you say same, you mean same? I mean, there are guidelines like same means change under I don't know 1%, for example. There are things like that, you know, to quantify what we mean same, what mean increase, and what mean decrease, and so on.
I understand those thresholds could be chosen. Yeah, I mean we could say we could orient it on RECIST. Yeah, what's clinically relevant? Yeah, certainly not 1 or 2%. Yeah, but also when you write your report, your oncological report. Yeah, I mean the feedback you always get from your oncologist is it's very nice if it has grown very little. Yeah, 1 millimeter here and 1 millimeter there, or if it has decreased in size very slightly. But does this justify a change in oncologic therapy? That's the question you want to answer. Yeah. And this threshold can be variable, to be very honest. Yeah, it can vary from the type of therapy the patient undergoes, and probably it can also vary from oncologist to oncologist. Yeah, and if not, we would look at criteria-based reporting similar to RECIST to standardize this approach. Yeah, but I cannot answer this question because in my point of view and my experience in working with our friends from oncology, this can be variable and also depends on the patient. Yeah, how old is the patient? What tumor does he have or she? So there are a lot of factors you need to take into account.
Okay, but there are no very much detailed guidelines for this.
No, because the parameter of total tumor volume is not established in clinical routine in a way that it would be established in guidelines for therapy guidance. Yeah, I think that's the major reason for it. And I mean, if you look at it, the pipeline will be cumbersome. Yeah, we need to develop the appropriate algorithms. We need to prove that the algorithms are performing and showing what they should. Yeah, then we need to validate them against established criteria like RECIST or immunotherapy against the iRECIST or whatever criteria you want to use. Yeah, then you need to show that it makes a difference and it has an impact. Yeah. So that's a very long story to follow, but I guess as always, if you don't start the journey, you'll never arrive.
Well, so thanks a lot to everyone who has been participating on this. So I guess we can agree that we really need to keep working together collaboratively and in actually establish this balance between the clinical effort for annotation, time, effort, and then the technical requirements implementation that are going to support the clinical specific needs. So thanks a lot for the discussion. I think it's been very, very nice to talk about this, and hopefully we can keep discussing in the different working groups and the different projects that where we are working. So thanks a lot.
Thank you.
Thanks.
Wonderful.
Thank you. Thank you for the interesting discussion and all the contributions from the others. I mean, we could discuss for decades here. There's a lot to do still, and yeah, this is always a starting point to go further.